Hello everyone, I'm Divya and today we are going to learn about how to solve crossing over questions which is from genetics. So many people they think that the questions related to crossing over are very complicated and it's very each to leave such questions during examination. But guys, believe me, these are the questions which will give you to score more in CSIR, JRF or GATE. But this type of question generally important for CSIR, JRF in life science. So it's very easy. Believe me, there is no need to bother about it. Just you need to know about some little tricks how to solve this question. And yes, today we are going to learn that. And after this, if you will find any such questions related to crossing over, then the distance between two genes, then I hope you will solve these questions out. But before that, we need to know what is crossing over. So crossing over generally terms entangled between the chromosome, that is called crossing over, which brings variations in the gene. So if let imagine this is maternal chromosome 1 and this is paternal chromosome 1, because in case of everyone there is pairing of each chromosome. So when crossing over will happen, then entanglement of chromosome happen and this will give you new variations in your gene. So and due to this variation, the result of such alleles change, like if you have one character A, B, C, three alleles, they perform one character. The same character will be in a different way because of change in one allele. Like this, if there are three allele and which perform same character, then how many different character you will get? That is 2 to the power n, that is number of allele, and that is 3, that is equal to 8. So there are 8 number of changes of characters you will find due to this variation. And there are only dominant allele or recessive alleles will be given in your questions, so be careful to that. It may be provided in the form of A, B, C, or in the form of characters name like short or height, like this, white or red. Like this, it will be mentioned that these are the two character, one would be for dominant and one would be for recessive. Like this, the questions will be provided to you. So let's come to question part. If any question, it will be asked like if the questions will be provided like this, either they will be provided in the form of character name, remember character name, either or in the form of allele. You just have to interpret in your own way. So when you will find such questions, just do one thing, just target the highest number you can see. Here 260 and 300, right? So just do one thing, 260 and 230, name them as parents. The highest number you will find, those are the parents. So the sequence of parents are ABC and ABC, the capital ABC and small ABC. These are the parent sequence. So in case of questions, there are many questions they will ask, like number of secondary crossing over right? Number of double crossing over and distance between two genes. There are many questions they can ask, and you can solve all the answers from this sequence. And don't worry, the number would be provided in your examination questions. So just you need to just remove out that which are the parents, which are the single crossing over, which are the double crossing over, and which are the nearest gene you can find out from your sequence. So guys, these, if this is parents, so A, B, C capital and small a, B, C, this is the parent sequence. There are no such sequence you can get. So this is parent sequence. And just find out like this, find out the smallest numbers, that is 20 and 40, right? This is, double crossing over. Okay, because single crossing over means if one chromosome is entangled for one time, but double crossing over is yes, so 1 and 2, and this is 1 but this is 2, so the chances of crossing over is less in this case, but the chances of crossing over is high in single crossing over, but double crossing over the chances are low. So the less number you will get, these are the double crossing over, and the remaining are single crossing over. Okay, SCO, single crossing over, double crossing over. So like this, you first have to remove out which are the double crossing over, which are the parents, which are the second crossing over, single crossing over. So when you will get these numbers, then you just need to formalize and you will get your answer like this. If when you will get A, B, C, this doesn't mean that the gene, when you will get A, B, C, this doesn't mean that these are the aligned form like this. This may be A, C, B. So you have to find out that A, C, B or A, B, C. So the question may be asked that A, B, C are the genetic sequence aligned or A, C, B. You have to find out from this question and you will get it very easily. So if you have to find out the distance between A and B, what you have to do? Just take parent allele configuration, that is capital AB and small ab. So the sequence which are not having capital AB or small ab, these are the target area for you. Like this, if you check AB, yes, A and B, yes, because capital, small capital, because in case of parents we have capital AB and small ab, but in case of this case you will get capital A and small b. Like this you can check, no, this is also parental, this is also AB capital, yes and yes, right? AB and AB. So just sum of all the value of the tick out mark, like first, second, third and fourth. 100, 140, that is 240, 20 and 40, 60. So 240 and 60, that is equal to 300, right? So just write down 300 divided by total number. if we'll sum up 100, 240, and that was 300, right? 300, 560, 860, and 860 and 8, 940 and 6, that is 1000 into 100, that's equal to 30, right? Like this, if we'll find out distance between B and D, we will get. Similarly, we have to finalize. Like, I'm really sorry I had written out D, B and C. Similarly, we have to find out the dissimilar sequence which are not in the parents one, that is capital BC and small b's. We don't have to find out this, we just have to find out the dissimilar sequence to this, like yes this one, yes this one, and yes this one, and yes this one, yes this one, and yes this one, right? So you have to sum this 100, 140, 240, and 300, 440. So that equal to 440 divided by 1000 into 100. To 44%. So that means B, C are lengthier than A, B, right? A, B and C. But sequence is A, B, C or not, we can find out only when we will count distance between A and C. So like this, if we'll find out A and C, yes, yes, and yes, yes, yeah, no, no, right? Yes. So 1, 2, 3, 4, 100, 100, 240, 100, 240, 240, 140, 340, 380, right? So the distance between AC 340 divided by 1000 into 100, that equal to 34%. So see, first one is your 30%, second one is your 44, and then 34. That means obviously A and C are the, B and C are the distant one and AC are the nearer one. So you can see A, C and B. No, AB distance was 30. Yes, so we get AB 30, BC 44, AC 34. So the easiest thing is when you will get highest amount, that are the longest distance. So B and C give the end point and A and the middle point. So your sequence should be BAC, right? So if the question will ask, multiple choice, there would be choices provided to you that BAC is the sequence or ABC is the sequence or ACB is the sequence. Like this you can also get the idea. So yes, like this you can solve what is the amount of SCO and what is the number of DCO, what is the number of parents, what are the number of total, and what is the sequence of the gene and sequence of alleles, and what is the distance between two alleles, or what is the longest distance between the end alleles. Like this multiple number of questions you will get from this numerical. So yes, if you will have any complications, please Please mention this in the comment section. I'll get back to you as soon as possible. And guys, yes, if you are preparing for CSIR JRF Life Science, so please don't forget to subscribe this channel for upcoming videos. I'm really sorry as I could not upload videos regularly because of my PhD work. As you know, I am a PhD student, so but I will upload a number of videos related to all the questions how to solve easily. So I hope you must have found some easy tricks how to solve crossing over questions of genetics. And yes, all the very best. Thank you.